Hi everyone, I'm Bao. I'm from Parkway Christian School. Thank you for coming again. So in my project, me and Candy, our overarching goal was to characterize HESC cells response to CpG. So CpG is our um, area of study. So before we get started with my presentation, I'm going to talk about a little bit about the key terms. So these are some terms that I think might be helpful to know. So we have human endometrial stromal cells, or in short, HESC and decidua. We also have CpG ODN, which is the treatment that we worked with. And I'm also going to talk about TLR-9 cytokines and CpG-PI complex. So again, our cell line is HESC, or human endometrial stromal cells. And as you can see here, this is the embryo where the baby is, and this is the decidua. So HESC cells are technically not part of the decidua, but after decidualization, it becomes decidua and endometrial stromal cells, which are part of the decidua. So you can see HESC cells over here. Talking a little bit about toll-like receptors. So they're a key element of the immune system, both innate and adaptive. Their role is to induce various cellular responses, including the transcription of several genes that later on leads to the elimination of pathogens like viruses and bacteria. So CpG, which is what we worked with, is recognized by TLR-9, which is an intracellular receptor. So this is the TLR-9 pathway in immune cells. So you can see here, after binding with CpG and undergoing phosphorylation, the binding of CpG to TLR-9 could induce either interferon alpha right here, or pro-inflammatory cytokines like TNF-alpha, IL-6, IL-8. So you might wonder, for those of you who do not know what cytokines are, so they are proteins that are released by the cells, and their job is to affect the interactions and communications between cells. So you can see in the picture over here, you have different cytokines and how they affect the communications of different immune cells, some of which we know kind of well, like B cells and T cells. So we're particularly interested in two kinds of cytokines, inflammatory cytokines and type 1 interferon cytokines. So inflammatory cytokines, like these three, are the ones that cause inflammation, which is the process by which your body removes pathogens. But you know from your doctors that too much inflammation could cause damages to your organs. So these cytokines need to be controlled by other cytokines. So that's when these cytokines come in. So for type 1 interferon cytokines, we have interferon alpha and interferon beta. They mostly, but not all the time, protect the host against pathogens without damaging the host. So now we're getting to CpG, which is what we worked with and our main area of study. So it stands for CpG oligodeoxynucleotides. It is a short single-stranded DNA, bacterial DNA, and CpG stands for 5' cytosine phosphate guanine and 3'. Again, it is recognized by TLR-9, and the one that we worked with is CpG class B. So before we get into the data, I'm going to talk about PEI. So we added PEI to form the CpG-PI complex because, as you can see over here, when you only add CpG to the cells, it only induces IL-6. But when you add the complex, it also induces interferon alpha. So that's why we used CpG-PEI. So far, we know that in immune cells, the NF-kappa B pathway, which you can see over here, is the dominant pathway in immune cells. But our hypothesis is that the interferon beta pathway, which is this, is the actual dominant pathway induced by CpG in HESC cells. So this is what we were trying to prove. So we designed experiment 1 to test whether or not our hypothesis is correct. So we treated HESC cells with 100 nanomolars and 400 nanomolars of CpG-PEI. And then we did RNA extraction to obtain the RNA of the cells. And then we did qPCR to measure the RNA expressions of the genes that we want to test. So as you can see in the picture right here, there are a lot of dead cells in the 400 nanomolar treatment group. So from this, we conclude that 400 nanomolar CpG-PEI is actually toxic for HESC cells. So these are our qPCR results. So as you can see right here, interferon beta is very upregulated, and this is our hypothesis. So these are the downstream interferon genes of interferon beta. So you have MIX1 and Vibrant, they're both upregulated. So we suppose this could be the result of the upregulation of interferon beta. So, and these are the qPCR results of the inflammatory genes. So we have TNF-alpha, IL-8, and IL-6, both upregulated, but when compared these genes to interferon beta, their upregulations are not as significant as what we saw previously. So our conclusion is 400 nanomolars CpG-PEI is toxic for the cells, as we saw in the pictures. And we were able to validate our hypothesis that interferon beta is the dominant pathway in HESC cells. And we also know that the effects of CpG-PEI on HESC cells are best observed at the 48 hours time point. So in our next couple experiments, our main goal was to verify the data that we got from the previous experiment. So this time we didn't treat the HESC cells with 400 nanomolar CpG-PEI because we know that it is toxic for the cells. And we did the same thing. We did RNA extraction and qPCR. So as you can see in these pictures, the cells are doing fine because this time we didn't treat them with 400 nanomolar CpGs. These are our attempt to verify the data that we got from the previous experiment. So you can see, again, interferon beta is very upregulated by about 30 times. And as the result, MIX1 and Vibrant were also induced by that. And when we get to the inflammatory genes, which are the downstream genes of the NF-kappa B, we could see again that they're upregulated, but not by that. So with this, we were able to verify that interferon beta is indeed the dominant pathway induced by CpG in HESC cells. So that is my project. So I want to spend a little bit of time to say thank you to Dr. Moore for his advice and inspiration, for correcting me for mispronouncing some of the scientific words. And most importantly, the opportunity to work in the Moore lab to gain so much knowledge about biomedical research. I thank Candy for her mentorship and her patience, as well as other people like Anna, Yuan, Hussein, Anthony, Andy for their insights and for their willingness to help me whenever I'm stuck and to answer my questions. I thank you, Alisa, for handling all the paperwork, and I thank Candy and Yuan for taking me to the lab and driving me home.
So thanks everyone.
Very good. Is there any question for
Are there any questions?
you don't have questions? You can ask. No? Okay, we'll let you go.
Thank you.